All right, we're going to continue on with DNA replication. Now we have looked at the Hershey-Chase experiments that helped to establish DNA as the heritable substance, but before we move on and look at the actual process of DNA replication, we're going to think about the uh, evidence that we have for how DNA is replicated. So this was what happened before we actually went through the individual steps. We had to figure out, how does it work? How is it being copied? And so there were three competing models for DNA synthesis after the discovery of the double helix. One was called conservative replication. There was also semi-conservative replication, and finally something called dispersive replication. And so experiments needed to be devised that could differentiate between these three competing models so that we could be able to appropriately interpret the results of the experiments done on the process of DNA replication. So here's the conservative model of DNA replication. Basically, what the conservative model says is that you have a parent cell that has two strands in each chromosome. So remember, from gene expression, the easiest way to say this, you've got a template strand and a non-template strand. So you've got two strands. And when these strands become replicated, basically the original strands that were found in the parent, they are conserved in a pairing. So they remain a double helix throughout replications. This means that basically they come apart, they're copied, and those copies go off on their own and make their own double helices. And this continues through each subsequent replications. Another way of saying this is that the parent strands reform and can then be reused. And so that's the conservative model. The semi-conservative model basically is a version of that, in which you still start with those two parent strands in the double helix. The parent strands then separate, and one of the parent strands is copied, and it has a new strand being formed, shown in light blue, and that parent strand stays with the new strand. And so it's semi-conservative because now in the daughter strands, each daughter strand has one of the parent strands within it. So each of those daughter chromosomes has one parent strand. And then as it continues, you see that the parent strand just keeps getting diluted. So now it's 2 out of 4. The next one, it'll be 2 out of 8, etc. And so this would be the semi-conservative model. And then the dispersive model, this one I laugh at a little bit. It was a possibility, but it's basically the idea that the daughter strands are basically just mixtures of the parent strands and newly synthesized DNA. And so in this case, I don't know exactly how this would work. It certainly doesn't work with our current model of how DNA is replicated. But at the time, it was a possibility based on the knowledge at that time. And so here we have another another pair of scientists, Mezels and Stahl, and they devised experiments that would test which of these three possibilities, conservative, semi-conservative, or dispersive, is correct. And so again, they're using E. coli, and in one case, they grew the E. coli with heavy nitrogen, and so it's going to be literally heavier than other nitrogen. And then there's also a light nitrogen, and so another set of E. coli was grown in a liquid that had that lighter version of oxygen. Since nitrogen is a part of nucleotides, that nitrogen is going to get incorporated into the DNA. And so they basically started with these two distinct parent strains of E. coli. One strain had light DNA, one strain had heavy DNA. And so they then crossed them. So they basically took the E. coli grown with heavy nitrogen and put it into the media with light nitrogen. And so the idea here is that the parents should remain with the heavy, but any daughter cells, because these E. coli are dividing by, they're basically just splitting in half. They're dividing by a version of mitosis. And so any daughter cell is going to have one of three versions. So remember, either they're going to retain that heavy, so there'll be cells that have just the heavy, but there'll also be about 3/4 of the cells that'll have only light, and that will keep getting diluted. So basically, if the conservative model is correct, if you let them divide long enough, you more or less won't see any of the heavy DNA by the end. It'll just be overtaken. There'll be an overwhelming amount of the lighter DNA from the light nitrogen media. If it's semi-conservative, more or less, you're going to see something similar. And then the dispersive model, you should see actually only the medium weight. So if it's mixing the heavy and the light, you're going to end up seeing just a medium weight for the DNA. So that's what's shown here. You can have either light, medium, or heavy, depending on what the results are. And so here is what their test was. They saw that after one round of cell division, in that taking the heavy DNA E. coli and moving them into the light nitrogen media, they saw after one round the DNA was medium weight. So this is just one round of replication, and hopefully you can see that that is actually consistent with two of these different models. That is no longer consistent with the conservative model. So in the conservative model, you should have two bands here. You should have a light band and a heavy band. Instead, what we see is a medium band, and that is consistent with either one of the strands of DNA in each DNA chromosome is going to be heavy and the other strand is going to be light, or it could be consistent with the dispersive model where it's just a mixture within. That could result in the medium. So that's after just one round. So if it was conservative, just make sure you can think through this. But the big idea is after one round, this is the first replication, you would have one DNA chromosome that is heavy and one that is light. So one heavy, one light. That is not what they saw. So now they continued on and they did 2 rounds of cell division in that new light media. And what they ended up seeing was now a mixture of cells that contained light DNA and cells that contain medium DNA. So it seems that the heavy DNA is gone. There are no more heavy DNA, so that eliminates, there was no maintenance of this original parental strand that was heavy. Everything now is either light or medium. And so is that consistent with semi-conservative or dispersive? If you look at dispersive, you'll see actually they're all still going to be about the same weight, so they should all still be medium if it's dispersive. Whereas in semi-conservative, that parental heavy strand is going to keep getting diluted out with every single replication. And that seems to be what we've seen here, since we have light and medium weight. So the light weights are the ones that are all new strand, and the medium weight is the one that has one parent strand and one newly synthesized strand. So this appears to be consistent with the semi-conservative model. So in this case, let's think about if it was dispersive, which DNA weights would be present after 2 rounds. So you can look at the illustration from your book and see that basically you're going to continue to have this medium weight, so it has a mixture of heavy and light nucleotides. So only medium would be the answer here. And so this is a video that you can view and try and maybe hear this in a slightly different way. I think they actually also add a few details to this, but the basic method is that the original strands of DNA contain nucleotides made from a heavy form of nitrogen, and anything that's newly synthesized would contain nucleotides made from a light form of nitrogen. So that is basically how we established that DNA replication is semi-conservative. So make sure that you understand what semi-conservative means and that this is the version that evidence supports.